One of the key features of object recognition is the distinction between what and where pathways. Let's start off, though, by considering the problem of perception. When people observe the world around them, they are never neutral observers. We see the world in terms of the threats and rewards that are relevant to our survival and our success. We need to be able to describe things and reason about them in terms of our goals, and we need to be able to operate things and use them. So these represent important problems for the perceptual process within the brain as we interpret the world around us. Perception is goal-driven and describes where things are, and this type of description involves positions, orientations, and actions, and it also describes what things are, and these types of descriptions involve features like colors and shapes. Researchers have found strong evidence that there are separate what and where pathways for visual information processing in both primates of all kinds and humans, who are also a type of primate. The ventral stream is involved in looking at what things are, and another stream that goes to the parietal lobe, called the dorsal stream, is responsible for determining where things are and for analyzing the spatial configuration between different objects that we are looking at. The pathways that carry visual information from the retina separate into two major fiber bundles in the primary visual cortex, also known as V1, within the occipital lobe. One of these pathways moves ventrally into the temporal lobe and is called the what, or ventral pathway, and the other pathway moves dorsally into the parietal lobe and is called the where, or dorsal pathway. One of the things about the way that the brain is organized is that similar things are not necessarily placed close together because the brain was not designed by an engineer. A good example of this is the fact that information coming into the eyes then proceeds all the way to the back of the brain at the opposite end and is processed in the primary visual cortex, and then in the case of the ventral stream, or the what pathway, goes all the way back close to the eye at the temporal pole and the inferior temporal cortex, where it is bound together in a form where objects are recognized. In principle, brain operations are describable in terms of wiring diagrams that show how patterns of connection and activation between different brain regions allow functions to be performed. The wiring diagrams for macaque monkeys in particular have been well described, and the corresponding wiring diagram for human vision appears to be very similar. In future decades, researchers believe that it may be possible to identify a complete wiring diagram for the brain, something which is referred to as the connectome. The information from the what and where pathways sends the processing of visual scenes to different parts of the brain, so that one part of the brain in the parietal cortex knows about the where information for an object, and another part of the brain in the temporal cortex knows about the what information. But in order to understand what objects are out there and where they are, we have to bind this information together. And this occurs in a process or a place called the convergence zone, which is in the inferior temporal cortex. So this convergence and binding of object information is really important. Otherwise, we'd only have these fractionated views of different aspects of objects and wouldn't be able to see them in a unified fashion. The proof that people have different pathways for visual perception originally came in research with monkeys and involved creating lesions in the parietal or temporal cortex and then showing that the monkeys had different deficits which corresponded to the idea of a distinction between what and where pathways. And this research goes at least as far back as the early 1970s, long before brain scanning and imaging was possible. Monkeys with bilateral lesions that disrupted their ventral streams had great difficulty discriminating between different shapes and failed to recognize what the object was, but they had no trouble determining where the object was in relation to other objects. And the opposite was true for monkeys that had lesions in their dorsal streams disrupted. So a Watt lesion occurs when part of the brain has been destroyed that affects the Watt pathway, and it gives us clues as to the capabilities that would normally occur in the lesioned area. Sometimes we can observe humans with lesions not because they've been operated on, but because they have some kind of trauma that's happened to the brain. In one well-known case, a woman got brain lesions after carbon monoxide poisoning, and in this case the lesion damaged her Watt pathway but not her Ware pathway. This could be demonstrated in a task where she was asked first to match the position of a card she was holding with a slot placed some distance away. In this case, she had trouble matching the position, but describing essentially what the object was. But when she could place the object as an action into the slot, she had no difficulty performing a task because the action task used the Ware pathway rather than the Watt pathway. And again, the lesions that the person had when they were imaged corresponded to the idea that the important parts of the Watt pathway had been damaged while the Ware pathway was intact. So there's plenty of evidence both through brain imaging of humans, through lesions in monkeys, and through knowledge of the wiring of the visual system to demonstrate that the processing of objects occurs through separate Watt and Ware pathways during object recognition. The Watt pathway goes to the temporal cortex and the Ware pathway goes to the parietal cortex. Somehow, the brain has to put all that information back together again so that it recognizes what the Ware and Watt information refer to the same object. This problem of getting the information back together again is referred to as a binding problem, and binding occurs in a convergence zone that exists in the temporal cortex.